The cell cycle is comprised of the G1 phase, S phase, G2 phase, and mitosis. Meiosis is another type of cell reproduction with similar step as mitosis. So how are they different? Mitosis is used for cell division and asexual reproduction, where the parent cell passes copies of all of its genes to the offspring. The daughter cell produced through mitosis contains the same genetic information with the same number of chromosomes as its parent. Single-cell eukaryotic organisms such as yeast and protists and some simple multicellular organisms utilize asexual reproduction. What are the advantages and disadvantages? Asexual reproduction is suitable for organisms that live in a stable environment and are unable to look for mates. Its disadvantages include the lack of variations between organisms, so if the living environment suddenly changes, the entire group can be wiped out. Then what happens when a complex multicellular organism, such as a human being, wants to reproduce? We cannot produce gametes, which are eggs and sperms, through mitosis because the process will create 92 chromosomes in the resulting zygote. Humans only need 46 chromosomes. The human body reproduces by creating gametes through meiosis. The purpose of meiosis is to reduce the amount of chromosomes in each gamete to 23. When an egg and a sperm fertilizes, the resulting zygote will have the complete set of 46 chromosomes. Now let's go over the stages of meiosis. During interphase, chromosomes replicated but are still uncondensed and invisible. At this point, each replicated chromosome must consist of two identical sister chromatids. Then the cell enters meiosis 1, where the homologous chromosomes will be separated. It begins with prophase 1. The chromosomes condense and become visible. This is the stage where crossing over occurs, which allows the genetic information to exchange between the sister chromatids. A process known as synapsis, where synaptomal complex forms to hold homologous together. Each tetrad had at least one chiasma where the crossing over occurred. Next is metaphase 1. Tetrads are arranged on a metaphase plate. Both chromatids are attached to a kintochore microtubules from one of the pole. Next is metaphase 1. Chromosomes move toward the opposite poles with the help of spindle apparatus. Sister chromatids still moves as a single unit. Afterward, the cells enters telophase and cytokinesis. Each half of the cell has a complete haploid set of chromosomes. Each chromosome is composed of two sister chromatids. Then cytokinesis divides the cytoplasm and forms the two daughter cells. In animal cell, a cleavage furrow is formed, and in plant cell, a cell plate forms. The cell is now prepared to go into meiosis 2. In prophase 2, a spindle apparatus is formed while the chromosomes are moving toward the metaphase 2 plate. During metaphase 2, the chromosomes are lined up along the metaphase plate. Sister chromatids are no longer genetically identical due to the crossing over in meiosis 1. Kintochores of the chromatids are attached to the microtubules extending from both poles. Anaphase 2. The centromeres separate and the sister chromatids come apart. They move as individual chromosomes toward the opposite poles. Telophase 2 and cytokinesis. The nuclei form while the chromosome deconsents again. Cytokinesis occur. So what are the results of meiosis? 4 daughter cells are produced and each has a haploid set of chromosomes. All 4 cells are genetically different than each other and the parent cell.